Okay, right. So in the previous video, right, so we stopped here in the I call it introduction to the reabsorption at loop of Henle. So remember, loop of Henle divided into two regions, descending loop of Henle and also ascending loop of Henle. So this is descending loop of Henle, right? And this is the ascending loop of Henle. Okay, you might be wondering something. Why is there a number? Is there a number inside the loop of Henle? So this number is actually representing the osmolarity of the filtrate, right? Ataupun the concentration of the filtrate inside the descending and ascending loop of Henle. Okay, all right, so we will discuss that later. Okay, now let's see what happened as the filtrate travels from the proximal tubule and then reaching to the descending loop of Henle. Okay, all right, so this is very important. The descending loop of Henle or descending limb loop of Henle is very permeable to water. So this is descending loop of Henle membrane, right? So the membrane of descending loop of Henle is very permeable to water because they have a lot of aquaporine. So aquaporine is the pore, right? Or channel that transport water. So there are a lot of aquaporine here along the membrane, which makes the membrane become so permeable towards water. So what happened? Water will be reabsorbed out of the filtrate into the surrounding capillary, all right? Or we call it as the vasa recta, or into the surrounding body fluid. All right, okay. So as that, you can see, as the filtrate loses water, right? So it will increase the solute concentration as descending extend into the medulla, right? So you can see here, right? Let me erase this one. You can see the concentration of the filtrate increase as the filtrate moving or transported or flow down to the descending loop of Henle into the medulla region. Ingat, medulla tu apa? Bahagian nephron yang belah dalam. Right? Dia ada cortex and medulla, right? So it become more concentrated as it reach deep into the medullary region. Okay, so you can see as water diffuse out via osmosis. All right, and the highest concentration of solute in the filtrate is 1,200, which is the concentration is similar to the concentration of tissue in the surrounding medulla. So maksudnya filtrate dalam ni jadi sangat-sangat pekat. Kita bayangkan mula-mula filtrate tu kan ada macam-macam. They have water, salt, amino acid, glucose and whatnot. But as it reach to the descending loop of Henle, water is reabsorbed. You just bayangkan air keluar dengan sangat-sangat banyak. So filtrate inside the loop of Henle become concentrated. So kurang air, right? So that's why you can see the numbers over here increase, which reflect to the concentration of solute that remain inside the filtrate. Okay, right. Okay, next. Okay, at ascending limb loop of Henle, okay, ascending limb loop of Henle has transport epithelium that lacks of water channel. So this one, remember, this is descending loop of Henle and this is ascending loop of Henle. So, I want to show you something. Look at the ascending loop of Henle. The ascending loop of Henle has two regions. The one here, right, we call it as the thin segment, right? And over here is the thick segment. Thin segment of ascending loop of Henle and thick segment of ascending limb loop of Henle. So this thin and thick segment actually reabsorb different solutes or molecule. Okay, right. But overall, the ascending loop of Henle is not permeable towards water, meaning to say at this region, there is no reabsorption of water. But what happened is just reabsorb salt. Okay, right. Okay, now let's see the thin segment first. Right, so this is the thin segment. Okay, so in the thin segment, NaCl will be diffused out of the permeable tubule into the interstitial fluid or into the surrounding capillary. Okay, the purpose is to maintain the osmolarity of interstitial fluid in the medulla. Alright, okay. Apa maksud dia tu? Tak faham lah, madam. Okay, tak faham kan? Okay, cuba tengok balik, ingat balik. Just now, in the descending loop of Henle, water is reabsorbed. So, the surrounding interstitial fluid become less concentrated ataupun has low osmolarity. Right? Air keluar dekat surrounding tissue tu. So, tisu-tisu kat situ, kawasan-kawasan situ jadi semakin ataupun kita panggil kurang pekat. Alright? So, this is not balanced. Right? Tak berapa balance. So, to balance the osmolarity, madam, kenapa nak maintain osmolarity? Okay. Inner medulla, remember I did mention just now, the inner medulla, kepekatan dia ialah 1200. Memang that is the nature of inner medulla. Kepekatan surrounding tissue or the surrounding interstitial fluid is 1200 milliosmol if I'm not mistaken. Okay, the unit. Okay, so as water is reabsorbed from here at the descending loop of Henle, so the concentration ataupun the osmolarity of the medulla become change, right? Maybe drop into 900 or maybe drop to 700, for example. So due to that, to maintain this osmolarity, dia nak kekalkan mesti lebih kurang 1200. So at ascending loop of Henle, it will reabsorb salt. So garam sahaja akan keluar. Right? So due to that, the surrounding interstitial fluid in the medulla will be maintained. Right? The osmolarity will be maintained. Alright? Okay? That's why you can see here, kepekatan dia pun, you can see as salt keluar. Now, the concentration inside the ascending loop of Henle will be reduced as more salts is reabsorbed. Okay? Alright. So, now, what happened at the thick segment of ascending loop of Henle? Okay. So, remember at the thin segment, NaCl is reabsorbed, okay, by passive transport, okay, from high concentration of NaCl to here in the interstitial area that have low concentration of NaCl. Okay, so at the thick segment, so the filtrate then reach to the thick segment. So at the thick segment, right, when NaCl concentration become decrease, right, so NaCl dah keluar kan, following the concentration gradient, so concentration of NaCl over here is decreased. Okay, so when NaCl concentration decrease at the thick segment, the epithelium will be actively reabsorb NaCl. That's why you can see it still reabsorb NaCl, but via active transport, right? So at the moment here, NaCl concentration in the medulla is increased in the surrounding interstitial area or interstitial fluids of medulla. Okay, that's why you can see at the thin and the thick segment, they have different modes of reabsorption of NaCl. All right, okay. So filtrate loses salt, but no water. And that's why you can see filtrate become more dilute. So it is, what you call it? You can refer to the number ataupun concentration of the filtrate. So the concentration of filtrate become reduced, meaning to say it become more dilute as it move up to the cortex in the ascending loop of Henle and then going into the next excretory tubule, which is distal tubule. Okay, Okay, so now let's see overall movement of water ions across the proximal descending loop of Henle, ascending loop of Henle. So remember what happened here at the proximal tubule, all right? So these are the components of filtrate, which include water, salt, bicarbonate ion, hydrogen ion, urea, nutrients, and also maybe some drugs, right? So these are what happened. This is what happened at the proximal tubule. So we have discussed the active reabsorption and also passive reabsorption that happened in the proximal tubule. And you can see the filtrate then move along the descending loop of Henle. This is cortex region. This is outer medulla. This is inner medulla. So remember, inner medulla concentration is, or the osmolarity is very, very high. It is very hypertonic. Ataupun kita panggil hyperosmotic. Alright, around 1,200. Tak apalah, kita sebut hyperosmotic pun cukup lah. Very hypertonic pun boleh. Alright, okay. So you can see as as the filtrate move down to the descending loop of Henle, only water reabsorb, okay, out of the filtrate or out of the tubule into the surrounding interstitial area or surrounding body fluid or the surrounding capillary. All right, and due to that, you can see what happened here. Filtrate become concentrated. Concentrated of the filtrate increase. Jadi pekat sebab air keluar because of a lot of aquaporins, right? Which assisting the reabsorption of water. So, and then as the filtrate move up to the thin segment of ascending loop of Henle, you can see that here there is reabsorption of salt passively. All right, and then concentration of the filtrate will become less. Kepekatan dia akan berkurang, right? Because salt is reabsorbed. As it goes up to the thick segment of ascending loop of Henle, you can see NaCl reabsorb actively because concentration of NaCl here is lower. Okay, all right. Again, why do we need to, the nephron need to reabsorb salt at the ascending loop of Henle? That is because to maintain osmolarity of the medulla. Osmolarity of medulla. All right, and that's why as it goes, the filtrate flows upward to the thick segment of ascending loop of Henle, concentration of the filtrate, all right, become kepekatan dia bertambah atau berkurang. So the concentration of filtrate is decreased, right? Sebab salt is reabsorbed. So filtrate become dilute. Okay, right. So next, we can see also here to note something. This is about secretion. We are not yet discussed about secretion because we are still discussing about reabsorption, right? But just for your notes, right? So we have also the process of secretion, okay, where the secretion is the process where the blood capillary or the surrounding tissue secrete solutes or molecule like hydrogen ion and potassium ion back into the tubule, right? To regulate ataupun the purpose is to regulate blood pH and also impulse transmission. Right? So let's say if there is excess hydrogen ion, for example, right? So blood is so acidic, so hydrogen ion from the blood will be secreted back into the tubule so that it can help to reduce hydrogen ion in the blood. Thus, it can help to regulate or balance pH of the blood. Okay, right. So we will discuss that later. Okay, next, before we discuss what happened to the distal tubule, now let's see the concept of counter-current system, right? So the counter-current system is actually involving the descending loop of Henle, the interaction between loop of Henle, descending loop of Henle, and also ascending loop of Henle. So the loop of Henle and surrounding capillary act as a type of counter current system, right? Where it will generate a very steep osmotic gradient between the medulla and the cortex, right? Dia akan tolong menghasilkan perbezaan kepekatan di antara cortex region and also medulla region. Okay, it involves active transportation of NaCl, right, with expenditure of energy, which is using active transport, right, from the filtrate in the upper part of ascending limb, which is at the thick segment, right, of the loop to maintain a high salt concentration in the interior of the kidney. In the interior of the kidney here is referring to the medulla, right? So salt is reabsorbed is purposely to maintain the interior of the kidney or to maintain the osmolarity of the medulla, right? So which enable the kidney to form concentrated urine. Okay, right. So it expend energy to create a concentration gradient that we call as counter current multiplier system, right? So you can see descending loop of Henle reabsorb water. Ascending loop of Henle is reabsorbing salt. So this process is actually help to maintain the gradient between the what call it cortex and medulla, and also the gradient of the medulla itself, right? And also assisting in producing concentrated urine. Okay, right. So this is the mechanism of counter current multiplier system. Okay, let me draw the loop of Henle. Okay, so assuming this is descending loop of Henle, okay, so this is proximal. This is distal tubule, right? So here we have the descending, and also this is the thin segment of ascending thin segment, and this is the thick segment. Thick segment of ascending loop of Henle. Okay, right. Okay, now let's see. So remember the mechanism of counter current multiplier system involving ascending and descending limb loop of Henle, right? So filtrate, bahan-bahan tertapis will flow in the opposite direction between the two limbs, right? So as the filtrate, okay, filtrate flows along down to the descending loop of Henle, concentration of the filtrate is increasing. Okay, concentration of filtrate is increased. Why concentration of filtrate increase? Because water is reabsorbed out at the descending loop of Henle. So filtrate concentration is increased at the descending loop of Henle, right? But as the filtrate move along the ascending limb loop of Henle, concentration of the filtrate will decrease. All right, okay. So thus, the longer the loop of Henle, lagi panjang loop of Henle, right? Kenapa lagi panjang? Because there are some, our kidney is actually distributed, has two different distribution of kidney. Ada dua jenis nephron sebenarnya kita ni. Satu, juxtamedullary nephron, and number two is cortical nephron. So these two is differentiated by the length of loop of Henle. So ada nephron yang loop of Henle dia panjang, ada nephron yang loop of Henle dia pendek, right? Okay. So the longer the loop of Henle, the longer the region of interaction between descending and ascending. Thus, greater total concentration of medulla can be achieved. So lagi pekat renal medulla. Remember, renal medulla yang mana ni? Kawasan yang tepi-tepi region-region ni. So we call as renal medulla. So lagi panjang loop of Henle, lagi pekat kawasan medala because you have more reabsorption of water at descending, and thus you have a lot more reabsorption of salt at the ascending loop of Henle. Thus, it will create steeper osmotic gradient between the filtrate and the renal medulla, right? So thus, it will be easier for the water to move from the collecting duct, ataupun it is easier for the movement of water from collecting duct, right, to the renal medulla and to the surrounding blood vessel. What does it mean? Okay, so maknanya apa? Okay, let me draw. Let's say this is our collecting duct. We are not yet reached to that particular part, tapi tak apa. Okay, so this is collecting duct. So as we have a very steep concentration gradient, so contohlah kawasan medala ni sangat-sangat pekat, very high concentration. So what happened? They are easier for the water here inside the filtrate to be reabsorbed out of the collecting duct. All right. Sorry, from the collecting duct into the renal medulla region and then into the surrounding blood vessel. So lagi banyak air akan diserap keluar from the collecting duct into the surrounding blood vessel. So that's what happened to your urine. Bila air lagi banyak keluar from collecting duct, your urine become concentrated. Okay, so madam, apa kesan kalau urine concentrated? Maknanya your kidney has reabsorbed a lot of water to your capillary, to your surrounding body tissue for the usage of your cell, right, of your body, right? So as a result, the urine produced, it will be concentrated. Okay, right. Okay, so this is what you can see, the picture of counter current multiplier system, and you can see the difference in terms of the concentration of filtrate as water reabsorbed at descending loop of Henle, and you can see this is the tip loop of Henle where the concentration of the filtrate is similar to the concentration of medulla. Osmolarity of interstitial fluid, kepekatan interstitial fluid dalam kawasan medulla. Okay, right. Okay, next. And you can see as salt reabsorb, concentration of filtrate will be decreased inside the descending loop of Henle, right? So as your the concentration of the interstitial fluid around medulla is very, very high, ataupun have high osmolarity, so it will become easier for water to be reabsorbed at the from the collecting duct to the surrounding tissue or to the surrounding blood capillary. Okay, right. Okay, now let's see what happened at the collecting duct. Okay, reabsorption at collecting duct. So collecting duct has transport epithelium, right? And which is the aquaporine. So meaning collecting duct also do have aquaporine. Sorry. Okay, contoh. Alright. So the reabsorption at collecting duct is actually controlled by hormone. It is controlled by hormone. So hormone control its permeability, ketelapan collecting duct. When kidney is conserving water. Contoh apabila buah pinggang kita cuba berusaha untuk conserve water. Nak jaga badan kita daripada kekurangan air, aquaporine will allow water to cross the epithelium. Right? Contoh bila kita puasa. Bila kita puasa, perasan tak? When you are fasting, right? So our urine will be very concentrated. Why? Our cell is very, during that time, our blood and our surrounding tissue has a very high osmolarity. Betul? Kawasan dalam darah kita akan jadi pekat, tisu-tisu kita akan jadi kurang air. Right? So increase its osmolarity. So as such, hormone, right, will actually control or increase permeability of the collecting duct towards water. So water will be reabsorbed. Air akan diserap daripada collecting duct to the surrounding blood capillary. Okay, so bila air masuk balik dalam darah, so our blood what we call it, blood osmolarity will decrease back to the set point, right? So tissue kita pun dapat air, so we can survive, something like that. Okay, so in the inner medulla, right? So this is inner medulla region, this is outer region, and this is cortex region. Okay, in the inner medulla, the collecting duct is permeable towards urea. You can see it is permeable towards urea. So urea diffuse out by passive, right? Passive transport into the surrounding interstitial fluid or into the blood capillary. So as urea is reabsorbed, remember urea is solute. So when solute is reabsorbed, so the surrounding tissue will become concentrated. Betul, dia akan jadi pekat. Osmolarity increase. Right? So what happened? Water usually will follow by osmosis. That's why you can see here urea and water together. Right? So when urea is reabsorbed, water will follow by osmosis to maintain the osmolarity. Okay, so reabsorption of urea along with NaCl over here, right? So NaCl is also solute, right? Which also contribute to the osmolarity of surrounding interstitial fluid, right? And it will contribute to the high osmolarity of the inner medulla, right? Which then cause water to be reabsorbed by osmosis. Okay, right. So water will be reabsorbed and thus filtrate will become concentrated. So as more water reabsorbed, right? So filtrate yang tinggal remain inside the collecting duct will be concentrated and urine that is produced will be hyperosmotic, or in other words, urine will be concentrated. Okay, right? Okay, so that is generally what happened at the collecting duct. Okay, but when producing dilute urine, contoh kita tak puasa, kita minum air go, banyak sangat air kita minum, right? So what happened? When producing dilute urine, collecting duct will be actively transport out NaCl, you can see, transport out NaCl, and the epithelium will be lacking of aquaporin. So aquaporin will not be functioning, right, or not activated. Thus, what happened? Urine that is produced will be hypoosmotic ataupun kita panggil less concentrated. Or easier word, dilute. Diluted urine. Okay, so urine that produced will be hypoosmotic to the general body fluids. Okay, right. So that is regulation that you can see when our body is conserving water or the kidney is conserving water, or when we drink or drank a lot of water. Okay, so next one we are moving into the process of secretion. Okay, the last part of this urine formation subtopic. Okay, so see you in the next video.